Well, good evening. Welcome. Thank you for joining us for the 2021 F.A. Gilfillan Memorial Lecture. Tonight, we'll hear firsthand how research findings in genetics can be translated and applied to practical questions in the field of conservation biology and the field of human health, and also how a career in the sciences can be influenced by technological innovations and chance encounters. The Gilfillan Lecture is a long-standing tradition in our college, one of the longest, in fact, bringing us together every year to celebrate outstanding science at OSU in honor of Doc Gilfillan, whose record of extraordinary science has positively impacted generations. Before the lecture begins, I want first to thank the members of the Gilfillan family who've joined us today for their generous gift that enriches our college, Oregon State University, and our community. Your contribution enhances our efforts to share scientific discoveries, cutting-edge research, and innovative teaching with our students, faculty, staff, alumni, and friends. We appreciate your establishment of the Gilfillan Memorial Award to honor Francois A. Doc Gilfillan, who served as Dean of the College of Science for nearly 25 years, from 1938 to 1962, and as acting OSU president from 1941 to 42. The award honors Doc Gilfillan's life and recognizes his extensive service and contributions to OSU and the field of pharmaceutical chemistry. His contributions are a lasting inspiration to all of us. Tonight, I'm honored to introduce Dr. Michael Blouin as our newest recipient of the F.A. Gilfillan Memorial Award for Distinguished Scholarship. This award recognizes faculty in our college who demonstrate a long and exceptional scientific career with nationally and globally recognized scholarly achievements. Our initial plans for a lecture last spring were delayed due to the onset of the pandemic. Mike is very well deserving of this prestigious award. In his 25 years at OSU, he's developed a remarkably broad, distributed, and well-funded research program that spans molecular ecology and genetics. A professor in the Department of Integrative Biology, Mike joined our faculty as an assistant professor in 1995 after teaching at Sonoma State University and the University of South Florida. His research in the field of evolutionary and population genetics spans the areas of biodiversity, of molecular ecology, and conservation. He has employed cutting-edge molecular and genomic tools to develop new insights into immune genes and pathways of genetic resistance to infectious parasites, host parasite genetics, and the causes and consequences of domestication in hatchery fish. Since 1991, Mike's research has been funded by 35 awards totaling over $12.8 million from agencies including the National Institutes of Health and the Department of Energy. He's published more than 100 papers in journals such as Science, Proceedings of the National Academy of Sciences, and Nature Communications. In the course of his career, he's advised 8 doctoral students, 12 postdocs, and numerous undergraduate students here at OSU. His former doctoral students and mentees have gone on to prestigious academic positions at other universities. In his talk, entitled "What Darwin Couldn't Imagine: A Personal Journey Through the Ever-Changing Field of Evolutionary Genetics," Mike will share his story as a scientist and will describe his recent work in health, disease, and conservation. This research encompasses salmon conservation in the Pacific Northwest and transmission of the human infectious disease schistosomiasis, which is a serious parasitic disease transmitted by snails that affects more than 250 million people worldwide. Mike, congratulations on this award. We look forward to the talk.
Well, thank you, Roy. I really appreciate it. Let me go ahead and share my screen and get started. Let me get this thing to display. All right, very good. Okay, all right. Well, once again, thank you very much. And I am truly honored to have this opportunity to speak to you, and grateful to the college and university and all my colleagues. And I'm going to talk about a little bit about my life and work and career, but I'd like to illustrate a couple things. And one is, as I look back over what I've done as a scientist, I'm astonished at how every few years there have been technological innovations which have allowed those of us in evolutionary genetics to just ask continually more interesting, complex, and expanding questions. And I've been along for the ride for quite a bit of that, and it's fun to go back and really see the changes. And the other thing I'd like to really focus on is that in any career, there are really important and influential people that you meet along the way. People who support you and give you chances, and it's really important to acknowledge them and that as you go along. And I'd also like to thank the Gilfillan family for supporting this award, and just point out to people, this was a super interesting guy. He knew a lot about a lot of fields. He spoke, I think it was 7 languages, including Chinook. There's a bunch of archival material about Doc Gilfillan available online and in the library, and I recommend people check it out. But I'm going to start the story a little farther back with Charles Darwin, who of course lived in the 1800s, and he published his seminal work, Origin of Species, in 1859. And here he is as a young man. He's all excited. He's about to go off and be a naturalist traveling around the world studying biology. And the key thing about what he proposed was not that evolution occurred, because that was pretty well accepted, but a mechanism, which is natural selection. And this was brilliant and of course true. But you know, you see pictures of him later in his life, and he always looks kind of sad. He's not smiling. And I wonder if it's partly because there was this huge hole in his theory at the time. And that was, they didn't have a mechanism for inheritance. They didn't really know how it worked. The best people could come up with was kind of a blending model where if you made a big one with a small one, then the offspring would be intermediate. And with that kind of a blending model of inheritance, you just can't get major evolution beyond the range of phenotype that already exists in the population, where phenotype means the characteristics of the organisms. And he went to his grave not being able to solve this major hole in his theory. And sadly, unbeknownst to him, a contemporary of his, Gregor Mendel, had figured it all out and published it in an obscure journal in 1865, just 6 years after Origin of Species. And Darwin never knew. It wasn't until 1900 that Mendel's work was rediscovered, and there was forehead slapping all around the world going, of course, this is how it works, and the field of genetics was born. And I'm just going to take a quick tour through advances. By the 1930s, population genetics was a full-fledged field. By the 40s and 50s, we knew about DNA. By the 1960s, we were able to, by visualizing enzymes in electrophoretic gels, realize just how much genetic variation there was out there in populations in the wild. That was a brand new revelation to population geneticists. And then from then on, it's just been an increasing series of new technological advances in our ability to look at DNA and compare individuals and populations. In the 70s, restriction enzymes were invented. The Sanger method of DNA sequencing in the 80s, that was improved. PCR was invented. The Human Genome Project was initiated. In the 1990s, microsatellite markers were discovered, which were a huge boon to population geneticists. These are highly polymorphic markers, meaning at each locus, at each gene, there are many different versions, many alleles. So these are great for things like parentage analysis and asking how different populations are. And then in the late 2000s, there was this huge game-changing technology, next generation sequencing, which allows us now to just sequence entire genomes, and this technology just gets better and better with every passing year. I mean, just to put it into perspective, the first human genome cost $2.7 billion with a B, and took 15 years using existing technology. We could now do, any of you watching this, here at OSU, we could do your entire genome beginning to end in about a week for a couple $1,000 bucks. At some of the big genome centers, less than 1,000. So I mean, this is just extraordinary to me. And I appeared here as a scientist in the early 1980s. That's when I began my scientific career. And I've basically been along for the ride at each of these steps. And for me, it's really fascinating to just go back and see what's changed. Well, I'll start a little farther back, talk a little bit about my family. These are my parents. My mom, she was from Puerto Rico, and my dad's from Texas. She was a linguist, and my dad was in the Navy. And my grandmother moved in with us when I was 10, and so I had the gift of being brought up speaking Spanish, so I speak Spanish really well. My dad was in the Navy, and so we lived overseas actually until I was about 10, and mostly in Argentina and Japan. So, and this is a picture over here of me, my sister Wendy, and my parents. And a couple things about my parents is they really supported my love of biology. I was a geeky little kid. I had fish tanks and tanks full of salamanders and frogs and snakes and stuff all growing up. And my parents totally supported it. It wasn't until later in life that I learned my mom hated that stuff. She was terrified of snakes. But to her credit, she supported me and knew that that was my passion. You know, I did all the usual things. I did science fairs growing up and so forth. But when I look at this picture, I also realized the privilege that I had growing up. I came from a middle class family, went to decent schools. My parents were able to pay for my college. And so, none of that was on my radar screen, the fact that I had the wind at my back. But I think about that now that I'm a professor and realize that a lot of students who come to us didn't have the same opportunities, and that it's really important to do what you can to ensure student success and bring everyone along. So, but I moved on from my middle school scientific achievements and went to the University of Virginia for college and got a biology degree. Then I went to Florida State University to get my PhD. And there I worked with a scientist named Joe Travis, who he studied life history evolution in vertebrates, particularly fish. And he was an excellent statistician, knew a lot about quantitative genetics, which is the field in which we use statistical methods to estimate things like, heritability, how much of the variation among individuals is owing to genes versus environment, that sort of thing. And since I loved frogs, I did a study on the quantitative genetics of larval life history traits and adult morphology, and asking whether selection in one stage could influence evolution in the other. And it was really fun work. After that, I moved just down the road to the University of Florida because my partner at the time, Sharon, had just started veterinary school. And I just showed up in Gainesville, Florida. I was like, hey, I'm a biologist. I need a job. Didn't know what I was going to do. And I wound up doing data analysis for the Fish and Wildlife Service. They had all these sonograms from manatees that they wanted analyzed to learn things like do males and females sound different. So that was fun. But then about 6 months later, I met this fella over in the veterinary school named John Dame, who I credit as being a very influential person in my career. And he was a molecular biologist. He was studying this parasitic nematode called Ostratagia ostratagie, which is a parasite of cattle, has a big effect, lowers productivity. And he was trying to come up with some genetic markers to tell northern populations from southern, because the ones up north have a different life history and, impact cattle differently. And he wasn't having any success. And so we talked, and I said, so has anybody just done a basic population genetic survey to ask how different the populations are around the range? And he said, no, not really. Never thought of that. And we talked some more, and he said, could you do that? I said, sure. And he had a year left on a grant. He hired me. I'd never held a pipetter in my life. They taught me how to do some stuff. And I wound up doing a just basic population genetic survey of populations of this parasite from around the US, northern and southern. And here we were using this ancient technique called restriction fragment length polymorphism, looking at mitochondrial DNA because it's a very abundant type of DNA. And here you cut it with restriction enzymes and you run it in gels and blot it and use radioactivity. It was pretty tedious, but we got the data and learned some really interesting things. One, these worm populations have just crazy high genetic diversity, like 10 times higher what you'd see in a vertebrate. And my conclusion about that was, well, these things just have enormous population sizes. Well, they can be tens of thousands of worms in a cow, and the herds of cattle are large. And then the other surprising thing was it looked like complete mixing among all these populations. Like the whole US was just one giant interbreeding population, which we call, and population geneticists would say, is high gene flow. And my interpretation of this was that, well, people just move cattle around, and moving the host is causing high gene flow on the part of the parasite. Now, this had some interesting consequences because this group of nematodes tends to evolve resistance to anti-parasitic drugs really fast. And this is actually really bad news for that, because with high genetic diversity, that means there's a lot of little mutations popping up all over the place. Kind of like with COVID now, as an RNA virus, it actually doesn't have a very high mutation rate, but it just has enormous population size. So it's continuing to explore all the mutational space. That's why we have these new variants appearing. The other bad news was with the high gene flow, that means if a drug resistance allele was going to pop up in one population, it's going to rapidly spread to others and get around the US very quickly. And so this was actually really interesting applied results of this work. Now, about this time, the University of Florida got a capillary electrophoresis machine, so that allowed us to do more of the same, but at a much larger scale. All we had to do was PCR. We get this beautiful sequence back. And so what I did then was a comparative study. I compared several species of nematodes, some that infect sheep, some that infect cattle, and some that infect white-tailed deer. And the hypothesis here was that nobody's shipping deer around, so we should see much more highly structured populations in the deer parasites. In other words, much less gene flow. And that's exactly what we found. The sheep and deer and cattle parasites look just like the previous species, and the deer parasite showed lots of genetic subdivision. And so this was good support for my hypothesis about host movement controlling gene flow on the parasite. Now this was fun and interesting work. I was learning new stuff, but I really had kind of like two take-home messages from this work. And the first was I liked doing applied work. My dissertation had been fairly basic science, but this had practical applications. And so I decided I wanted to focus the rest of my career doing as applied stuff as I could. And then the second one was, I was kind of surprised at the reaction to this work in parasitology circles. They thought this was the coolest thing they'd ever seen. Whereas for me, I had brought techniques and data analysis methods from one field, basically working with vertebrates, and brought it into parasitology, where no one was doing this sort of thing. Basically at that time, they were trying to like name them and kill them, and doing things like measuring gene flow and population structure was brand new. So I quickly became a big fish in a small pond, as it were, because no one else was doing this. So what it taught me, though, was the importance of bringing people together from diverse backgrounds when we're trying to innovate in science, and how important that is. So after this, I did a 2-year teaching gig at Sonoma State University and a 1.5 years at the University of South Florida before I finally got my dream job at Oregon State University. Now back then, our logo was a little simpler, and this was our mascot, Friendly Benny Beaver. And I've been here long enough to see him evolve into more and more fierce versions of the same species. Of course, I looked a little different back then too. This is the photograph of me that was on the bulletin board outside the Department of Zoology office back in 1996. And those glasses really make me cringe, but fortunately I moved on. So when I got to OSU, I had just started working with this new class of markers, microsatellite markers. And again, they're wonderful because they're highly polymorphic, lots of different alleles. So for example, these 3 traces show the genotypes of 3 individuals. This one has these 2 alleles, this one has these 2, all 3 of them share this allele. So this makes them beautiful for things like parentage analysis, matching, say, parents and offspring. For population genetic structure, how different are these 2 populations of the same species, estimating gene flow, estimating the effective population size, which is a number of a population. It's usually smaller than the number of individuals that predicts how fast it's going to undergo genetic drift and basically loss of genetic diversity. And about this time, I was contacted by a student from Louisiana named Charles Criscione, Now, he just finished a master's degree in parasitology and wanted to get into population genetics. And so he joined my lab, and he did a whole really interesting series of studies using parasitic trematodes of salmon. Now, trematodes are a type of flatworm, and they have these complex life cycles that usually involve sometimes like a vertebrate host. They all usually go through snails, sometimes other hosts. And Charles asked a whole series of interesting questions about just the basic population genetics of trematodes, and nobody had done this before. He asked questions like, how does host movement influence parasite genetic structure? How do you even define a parasite population? What's the unit of random mating? Is it the parasites in a host? Is it the hosts within a particular geographic variation? So this was all new stuff. And now parasite population genetics is a field, and Charles is one of the leaders in that field. About the same time, I also turned my attention back to my first love, which was amphibians, and started a couple studies on the conservation genetics of threatened frog species in the Pacific Northwest, in particular these three here. And this work was carried out over about a 10-year period by three graduate students, Kirsten Monson, who's now a professor at Montclair State University, Eric Hoffman, who's now a professor at the University of Central Florida, and Ivan Philipson, who is co-owner of an ecotourism company called Wild Latitudes up in Portland. And they asked a series, they did a whole series of papers over about 10 years, generally focused on conservation genetics, like how different are populations of the same species, what are their effective sizes. And I gotta say, this was really fun work, mainly because it involved field work. We'd go up and catch frogs in these beautiful scenes in the Pacific Northwest. It was a lot of fun. And like I said, they published a bunch of papers, but I'd say some of the most important take-home messages we learned from this of conservation consequence are that populations of these frogs in the Pacific Northwest are very small and very isolated from each other. And I think that's because of the habitat. They have these aquatic habitats separated by large tracts of dry and hospitable habitat. So for example, this shows a measure of gene flow between pairs of populations as a function of geographic distance. This is for the Cascades frog. And it just drops off precipitously after about 10 kilometers. Similarly, we measured effective sizes and number of populations of each of these species. They all tend to be less than 50, which is a tiny number. What this means, if your populations aren't connected by gene flow, they're going to start losing genetic diversity and inbreeding pretty quickly. So this is important information for managers. I'm particularly proud of this study on the Oregon spotted frog, in which this was mainly Ivan's work on this. What we did was we took the remaining populations of the Oregon spotted frog, which has become very fragmented in its current distribution, and we determined where all the major original genetic subdivisions were. And this was really important information that was used by the Fish and Wildlife Service when they finally listed the frog as an endangered species in the mid-2010s. So this was really fun work, and I did move on to other things, but the next thing I moved on to actually had its origin back in Florida when I was out in the dark catching frogs. And I remember thinking, how do they know who's who out here? What's the chance that two frogs are going to meet and they're relatives? And how do they avoid inbreeding? And at the time, I thought, wow, wouldn't it be amazing if we could like take a tissue sample and reconstruct the pedigree of a wild population, figure out who are siblings, who aren't. But of course, at the time, that was crazy. There was no technology to let you do that. But with the development of these microsatellite loci, I realized that, yeah, this is the ticket. Because when you have lots of loci with lots of rare alleles, individuals who share a lot of rare alleles are going to be relatives. So I did some work, some simulation and empirical work showing that actually, with just a relatively small number of loci, you could do things like tell full sibs from half sibs from unrelateds. And of course, nowadays, this sounds really mundane. Nowadays, you spit in a tube, you send it to 23andMe, and they tell you, you've got 40 third cousins. But back then, this was actually quite eye-opening because people didn't realize you could do this. And my dream was, well, let's go into a wild population of which we know nothing and just reconstruct the entire pedigree. Who begat who, who's related to who. And I was thinking about, where I could do this with a wild vertebrate population. And then it finally dawned on me, salmon, of course. They go out to sea and then they all return to the same spot as adults. So if one could catch them, one could get a DNA sample and start building this population-wide pedigree. And so I started asking around, and I was directed to Catherine Costa, who is a geneticist for ODFW, who also another very influential person in my career. And I approached Catherine with this idea and said, hey, is there a population I could work on and get tissue samples? And she said, yeah, that sounds interesting, but there's this other problem that people would find way more interesting. And here's the thing. A lot of fish are raised in hatcheries. They release, they come back and spawn in the wild if they're not caught alongside wild fish. And what we do know is the productivity of wild populations is negatively correlated with the number of hatchery fish spawning with them. And it's been suspected for a long time that hatchery fish actually have lower fitness than wild fish, but no one's actually ever really tested that. Could you do that with these techniques you're talking about? And I was like, sure. And she said, all right, I've got the place for you. And she set me up working in the Hood River, which is a medium-sized river coming down the north side of Mount Hood. And it's a beautiful study system. It has this nice population of steelhead that go upstream every year. And there was a dam blocking the river. And so every fish to go upstream was handled by the ODFW. They sexed it, they weighed it, they took some scale sample to figure out how old the fish was and then tossed them upstream. And the really nice part was they would pass all the wild fish upstream. By the way, hatchery fish are all marked, so you can tell which is a hatchery fish. And then they'd pass upstream an equal number of hatchery fish. And the hatchery fish they were using were what we call F1s. They were first generation hatchery fish created using 2 wild broodstock, because this was a supplementation program. They're trying to enhance the wild population. And so Catherine said, well, we'll start collecting fin snips for you. But it turns out that we could get DNA out of these old scale samples. And they had scale samples going back to 1990. And so we started working on them, and we were able to put together basically a genotype of every fish that went upstream over a 20-year period. This is just by matching parents with their returning offspring who returned as adults 4 to 5 years later. And so we built this 4-generation pedigree with which we've been able to ask a bunch of interesting questions. In fact, we published over 20 papers on this system on a variety of topics, but I'm going to focus on the hatchery wild part. And this is mainly the work of these folks I want to acknowledge in particular. Mark Christie was a postdoc with the project. He's now a professor at Purdue, and Hitoshi Araki was also a postdoc. He's a professor at Hokkaido University. And I also want to acknowledge all the help that I got from the Oregon Department of Fish and Wildlife. People at the dam, the people working at the hatcheries, the district biologists, people like Catherine. And I've found the agency to be incredibly supportive of this work, very interested in the science and knowing what the truth is, no matter where it leads. And this work would have been completely impossible without them. So I just wanted to really acknowledge that. So some of the key things that we found in regards to this hatchery wild situation, the first important paper was Hitoshi led in 2007. We published this in Science. And it showed that the relative fitness of hatchery fish, shown here on the y-axis relative to wild, it declined pretty fast with generations in captivity. And these 2 points are particularly interesting. They were both from the Hood River, but for hatchery fish created using 2 wild parents and fish created using a wild and a hatchery. And they differ in fitness by almost 2-fold, which is just a crazy, unbelievable result. Yet it was repeatable several years in a row. Because we had this long pedigree, we were actually able to look at the fitness of wild-born fish as a function of their parentage. And what we found was, again, wild fish that had 2 wild parents had like double the fitness of wild fish that had 2 hatchery parents. Here, C stands for captive in this graph. And then finally, Mark did a really interesting study showing that although these F1 fish have lower fitness in the wild, they make much better broodstock, producing almost twice as many returning hatchery fish than do wild broodstock. Also, when he compared among different wild families used as broodstock, some did well in the hatchery, some did poorly in terms of producing returning adults. But those that produce a lot of returning adults, those adults don't do so well in the wild. So there's this huge trade-off. You can either do well in the hatchery or well in the wild. You can't be adapted to both. And so this really brought home the problem for managers that they had been suspecting for a long time, is that these hatchery fish are really different. And so now they have to somehow segregate them from wild populations when you have endangered wild populations. And this causes problems because you have reduced harvest opportunities, reduced angling opportunities. So it's this real trade-off. And so now the question is, and I also want to point out that when we first put this stuff out, it got a fair bit of press, but a lot of pushback too. A lot of people did not like what we were showing here. But in the last 10 to 15 years, there have been maybe 7 or 8 other studies using other populations and other species, which pretty much find the same thing. So it's pretty much widely accepted now. And now the big question is, so what's going on and what can we do about it? In other words, there's clearly some type of really intense selection that happens in a hatchery, which perhaps isn't surprising because it's so different from a wild environment. And then the question is, well, what can we do about it? The problem is we don't know what traits are under selection in the hatchery, and we don't know what about hatchery culture causes that selection. If we could figure that out, then perhaps it might be possible to alter the hatchery environment in a way that reduces those selection pressures, slows the rate of domestication, and allows us to produce fish that are more like wild fish. This would have many advantages, and most importantly, that you would not have so many concerns about them mixing with wild fish. And so this has been the focus of part of my lab's work for about the last 6 or 7 years. And we've been doing most of this work at the Oregon Hatchery Research Center, which is it's a cooperative venture between Oregon State University and the ODFW. And it's a really neat facility. They've got a lab, they've got interior labs, offices, places to stay. And they've got all these tanks of different sizes that we can use to raise fish. They also have these artificial streams, which are piped with river water. And so you can raise your fish, say that you can create the same families and raise some of the offspring in semi-natural conditions and some of the offspring in hatchery conditions. So it's a great facility in many ways. And I'd like to acknowledge in particular 2 people who are instrumental to this work. The first is David Knox. He was the director of the OHRC from its founding, and he was very supportive of our work. He was the one who first invited me to work out there. And he was a great person to interact with. Unfortunately, last December, he very unexpectedly passed away. And so he's left a big hole in in this type of research and at the center. I also want to thank the various managers of the staff who've been there, Ryan Couture, Joseph O'Neill, and Jen Krajek, who've been very helpful in our work. And then the other person I want to acknowledge is Neil Thompson, who was my graduate student originally and then was a postdoc on the project. He's now a geneticist with the USDA. And he's the one who first got us started in this type of work where we're actually looking at, raising fish and measuring their growth rate and that sort of thing. And just to give you an idea of the type of questions we're asking, what we're trying to do is figure out what kind of conditions we could manipulate about hatchery culture that would reduce the variation among families and their growth rate. Now, and they're basically their size at release. And you might think, well, why would you want to do that? Well, we know that larger smolts, those you release, have better survival at sea. We know that families vary in their ability to grow quickly in a hatchery, and that's owing to some traits we don't know, maybe their behavior, their physiology. So what happens is you wind up selecting in the hatchery for whatever those traits are that allow some fish to grow fast. And so we are trying to find conditions that reduce that variation. We're also trying to understand the traits under selection. And here's the thing. I mean, it's not that growth is bad, but how you achieve fast growth is different depending on the environment. In the wild, there's this trade-off between growth and survival. You have to, optimize the amount of energy you spend, fighting for a territory, foraging for food in the presence of predators, et cetera. Whereas in the hatchery, all you got to do is just eat and get big. And so what I think is happening is the hatchery is selecting for extreme values of traits that are genetically variable in the wild. And then when those fish come back, they pass them on to their offspring, and they don't survive, and that's why we see the lower fitness. So some of the traits we've been studying involve behavior. And in these experiments, for example, we'll create a bunch of full SIP families, raise them together in tanks, and sort them out at the end to see which family grew fastest via DNA. Then we look at the behavior of each family. So for example, these are racks of 12 tanks, and we have video cameras in front of each, and there's an individual fish in each tank, each from a different family, and we're measuring various aspects of their behavior. In particular, we're studying whether hatcheries inadvertently select for bolder behaviors. We know this kind of shyness, boldness axis is highly heritable. And this work has been done by Miriam Cameron and Mia Ray, who were a postdoc and a student in working with us for a while. And I also want to point out that basically some gratitudes to the John L. Fryer Aquatic Animal Health Lab, where we do a lot of this behavior work. Ruth Milston Clements is a super collaborator. She's the manager there, very supportive of our work. She's got great staff. This is Tim Summers and Stephen Fogarazzi. Here they are helping measure some fish for an experiment we took down. So I really wanted to thank them. And so we hope to continue doing this type of work, and we hope in a few years maybe have some recommendations to the agencies about how they might alter the way they raise fish. And so finally, the last kind of chapter is some work I started in the early 2000s with my colleague, Chris Bain. Now, Chris is now retired. He now happily lives in Arizona. But when he was at OSU, he was studying Schistosoma mansoni, which is another trematode, but one that parasitizes humans. And it uses these snails in the genus Biomphilaria as their intermediate host. And it's a really bad disease. It's in tropical regions. It infects hundreds of millions of people, kills hundreds of thousands each year. In terms of parasitic diseases, it's second only to malaria in terms of basically disability adjusted life years, morbidity, mortality, that sort of thing. So it's a really surprisingly important neglected tropical disease that a lot of people haven't heard of. And the way you get it is by being in water where the snails are shedding the parasite. This is a parasite species that occurs primarily in Africa. And the way it works is it infects the mesenteric veins of humans. The eggs are passed out in the feces. They hatch into a little swimming parasite when they hit water, infect the parasite, which then releases thousands of little parasites that go swimming around looking for a human, and the cycle starts over again. So following in Chris Bain's footsteps, we are focusing on the snail stage of the life cycle as we think the really vulnerable stage where you could really break the cycle of transmission. And the reason to focus here is that, one, there's only one drug that effectively treats this disease in humans. Now, it'll treat the disease, but people then just tend to get reinfected fairly quickly. Also, there's concern now that drug resistance is evolving in some populations of the worm. Also, now you can actually control the disease by getting rid of the snails. That's super effective, but it's one, difficult and ecologically damaging. You know, draining wetlands or spraying molluscicides, which is the preferred method nowadays. So our idea is, you know, and this is way off in the future, and right now we're doing exploratory work, but our idea is that if you could learn enough about the genetics of resistance to the parasite on the part of the snail, then you might be able to genetically alter snail populations so that you could have snails in the wild that are functional parts of the ecosystem, but that are unable to transmit the disease. And so this would perhaps be a long-term application of our work. And again, I started this with Chris. Through Charles, I got interested in trematodes. And Chris and I were always friendly. And I always thought this was a great study system because these snails, they're aquatic. You can raise them by the hundreds in fish tanks. You can have, pedigreed lines of snails. And you can challenge each individual snail with a certain number of parasites. And then a couple weeks later, you can ask whether this snail is infected or not. And if so, how many parasites is it shedding? So for example, this is a snail in a Petri dish here that's shedding parasites. Each of these little white dots here is a trematode swimming around looking for a human to infect. So it's really a neat study animal. And with Chris, we developed a panel of 50 inbred lines that ranged from low to high resistance. And we were testing various candidate genes that Chris thought might be involved in resistance. And I want to acknowledge former student Caitlin Bonner, who was instrumental in a lot of this early work. She was helping study in particular one of these candidate genes. And so we were working with Chris on this work, and he basically retired right around the time in the late 2000s where this game-changing technology came onto the scene, and that was the development of next generation sequencing. And here you fragment the total genomic DNA into little pieces and just sequence all of these millions and millions of little pieces in this massively parallel way. And then you have a computer line them back up based on their overlap. And with this, you could now, resequence entire genomes. And it immediately became apparent to me that this was the ticket, that we could use this technology to rapidly scan the genome of snails to find regions in which there's allelic variation controlling resistance. And so that is exactly where we took it. So our work for the last, since then has been mapping and characterizing resistance genes in Biomphilaria. And some of the key people involved in this work are Jacob Tennyson, who actually was my student originally. And then he went off, did a postdoc in human genomics, and then he came back and worked with me on this project. He's been the statistical and bioinformatics brains behind this work for many years. He's now moved on to studying mosquitoes and malaria at Harvard. Stephanie Blouin actually got her PhD here at OSU. She took population genetics from me back in the 90s. And she's a faculty research assistant who's been working with me for over 6 years now. And then finally, Ewan Allen's an immunologist who joined us for 3 years, and he's now a professor at the University of Nevada, Reno. And we've done a variety of things with this system, but I'd just like to kind of walk through one particular example of stuff we've done. And this starts with my colleague in France, André Theron at the University of Perpignan. And he had this population of snails from the island of Guadeloupe in the Caribbean, where schistosomes also were cycling. And he found that only half of the snails would get infected when you challenged them with parasite. And that to me smacked of genetic variation. So I talked him into doing a selection experiment where he took his population, challenged them, and kept just the ones that didn't get infected and took their offspring, started the next generation, and did this for 5 generations. And we saw a huge response to selection. What this shows is that the base population stayed at 50% infected over 5 generations, whereas the 2 selection lines went down to less than 10%. So highly heritable trait. So what we did was we took pools of individuals from each of these 3 groups, did genome scans using this new technology, and we found one region that just popped out like a sore thumb as having changed dramatically in going from the base population to the selection lines. And what we found was this 400,000 base pair region that had about 20 genes in it, one of which conferred resistance and the other 2 susceptibility. This just shows that the R allele was completely dominant over the other 2. If you have at least one R allele, like 25% get infected. If you don't, 75% get affected. So it's a really important gene, whatever it is, of the 20 in there. And in the middle of this region where you're able to analyze it, and a computer will identify protein coding regions and their putative function. And what we found was there are some genes which appeared to be what we call single pass transmembrane spanning regions. And these are proteins which stick inside cell walls. Here's a cell wall, and part of it hangs outside the cell interacting with the environment. The other part stays inside and signals. And so these were almost certainly the genes involved, especially since the outside bit was highly, highly variable between the 3 versions. And so Ewan did a whole series of studies. I think he had like 7 papers on this, studying these genes. And he had two main, I think, most important results. And one, he used this technique called RNA interference to knock down expression of the protein, basically to reduce the protein amounts. And he found that that caused the snails to shed more parasite. So that proved that these proteins were involved. This just shows the controls versus the knockdown snails, and this is the number of parasites shed. The other neat thing he found, which was even more intriguing, was that the different genotypes had different microbiomes. Now, snails have an open circulatory system, which is full of bacteria, and that's what the parasite first meets when it penetrates the snail. And so what this result suggests, here TM means these transmembrane genes, what this suggests the possibility that maybe rather than interacting with the parasite directly, maybe what these genes do is they somehow modify the microbiome, and the microbiome is either more or less hospitable to the parasite. So this opens up a whole new area of research that neither us nor any other groups have pursued yet. It's still just sitting there. So I hope maybe we can work on that in the future. We continue to do what's called genome-wide association studies, finding new regions, trying to characterize the genes. Like right now, we're studying this one region that has, again, interestingly, 3 different versions, but it's a crazy region. There's huge insertions and deletions when you compare the chromosomes. Some genes are only found on one chromosome and not the others. And I have a postdoc, Blouin, who's currently trying to get RNA interference working to knock down these genes to, again, test to see which is causal. So we hope to continue doing this type of work. I hope learning more and more about the genetic regions involved in resistance and what those proteins do. And again, ideally, at some point in the future, we will be able to turn this into some practical application for how to break the cycle of transmission. And so I'll wind it back to Charles Darwin, who I think if he could come back today and look at what humans have done and how far they've taken his theory in genetics, I think he'd be both astonished and delighted. And I think in this photograph, there'd be a big grin on his face. So that makes me happy. And finally, I'd like to end this by acknowledging all the many people who I've worked with over the years. This is Stephanie again, and Kate Paramislava, who was a fantastic technician who worked in my lab for several years. Way too many people to possibly put on one slide. This is just a sampling. And of course, I need to acknowledge the funding agencies. NIH has paid for all the snail work, and Bonneville Power Administration, ODFW, for the salmon work. And finally, I'd like to thank my family. My son Avery, my partner Sarah, her daughters Maya, Ellie, her son Sam, and her daughter Nicole. And I think it just kind of goes without saying that for people to be successful at work, it's very important to have a supportive family, and I have that family. And as fun as this stuff is, at the end of the day, it's nice to just set it down and go home and be with the people you love. So I want to thank them as well. And with that, I am happy to end this and take questions.
Well, thank you so much, Mike. That was wonderfully fun to hear. I learned a lot. I think a lot of us did in the audience. I'm Gabs. I use they, them pronouns. I work in the College of Science doing student engagement work. And I sometimes get the opportunity to share some time with folks and ask good questions. So one of the first questions that came up in the chat is from Chris Bain. I'm going to go ahead and answer that live here, and then I'm going to put it in the chat for you, Mike. But the question is, epigenetic effects were becoming mainstream, excuse the pun, about when you were publishing your work on hatchery effects in salmonoids. Has it been found that the heritable epigenetic changes have meaningful consequences in fitness changes in these populations?
So talking about the salmon.
Yeah, there's quite a few salmon questions. So just get your brain in that space.
People are most interested in the salmon stuff. That makes sense, given where we live. Yeah, actually, we actually did a little work looking at epigenetic changes between hatchery and wild fish. And we were using a very crude technique at the time and did not discover any meaningful differences. But in recent years, other people have done that and have found differences. So there's something about the hatchery environment does cause epigenetic changes. Now, linking that between that and specific fitness traits or fitness in general has yet to be done. But it's a really interesting area of work for sure.
Well, I've got another question on the salmon. So how do you separate hatchery raised fish released to the wild from wild fish? Do you have
Yeah, the hatcheries do
information
that.
about
It's incredibly laborious, but they actually cut the adipose fin off every fish they release. That's, you know, hundreds of thousands
Wow.
of fish. The adipose fin is this tiny little fin on the back that whose function is kind of unclear, but they seem to survive pretty well if you cut it off. So all these hatchery fish are marked. So that's how when they come back to the dam, the people working the trap know which is a wild fish and which is a hatchery.
That's amazing. Another question. Ben says that he's read some recent papers looking at variations in DNA methylation between hatchery and wild salmon. Might these differentially methylated regions be related to some of the behavioral differences you and others have observed?
Quite possibly. But what's interesting about this work, when we first started this work and we found that hatchery fish were different from wild, we thought, okay, well, there could be a variety of explanations, you know, epigenetic possibility, you know, maybe just generalized genomic deterioration because you have a huge relaxation of natural selection. So it could just be mutation accumulation. But Mark's work showed that what's going on is actual adaptation to the hatchery, which is a very different thing than just kind of genomic being messed with. And so my belief is this is just plain old Darwinian selection, albeit incredibly intense, strong selection that's causing very rapid evolution. And I don't think we need to appeal to any more exotic genetic explanations. It's just, I mean, people have been domesticating animals for thousands of years, and it's always the same story. So I don't see why fish would be any different, except this blistering speed at which it happens in this situation is the only thing that's surprising. So that would be my opinion on that. But there's definitely a lot to be done with epigenetics and seeing how that might be involved.
Well, let's move on to the snails. We've got a couple of really fun snail questions. So what do you mean by challenging the snails with parasites? Does this just mean introducing the parasites to the snail to see if the snail is infected or resistant?
Yes, exactly. So I showed that 12-well petri dish that had little snails in it. And you have a snail and you add 5 parasites to each well. And you let the parasite do their thing, and then you check them 5 weeks later to see which have gotten infected or not. So that's the challenge, and the snail either fights it off or they get infected, and that's kind of the binary response that we quantify.
And then does Esmosoni harm the snails? Also, are the snails infected at different rates depending on their age?
Yes and yes. Actually, it's a castrating parasite. It eventually makes its way to the ovotestes, and basically what it uses the snail for, it's a parasite factory. It basically, it starts replicating thousands of times and eats up all the ovotestes, which sterilizes the snail. And then all the parasites come out. So yes, it definitely harms the snail. And what was the second part of that question?
The second part was, are the snails infected at different rates depending on their age?
Oh, yes. Yeah, actually, it turns out that younger ones are more susceptible. There's kind of a
Interesting.
transition period. So when we quantify resistance in, say, an inbred line or a population, we're always very careful to kind of choose a particular size and have all our inference be about snails of that size.
That's so great. I do want to bring us back to the hatchery. Just a couple more questions about that, if that's okay. So Bob Mason here in the audience has a question that is this hatchery effect seen all over the world? And if so, is there variation observed that might lead to insights into what is going on over here?
You know, that's funny because most of the studies of this type have been done in North America. There's been a few on European brown trout. So I guess is the question. So is there anything we can learn by different results among different studies? Well, for sure, one result is that the older and more highly domesticated the salmon stock is, the lower the fitness. That seems to be pretty consistent. But now there have been, like I said, there have been, you know, close to 10 studies all looking at early generation hatchery fish, and they kind of all find the same thing. So yeah, it seems to be a pretty universal phenomenon.
So I have a question. I'm curious about teaching and your experience teaching at OSU throughout your time with us, and how teaching has changed over the years that you've been here, and how you've experienced that, or maybe how students have changed. Yeah, what are your thoughts on that?
Actually, that's really interesting. When I went through graduate school, nobody taught you how to teach. You just winged it. And I just kind of made it up as I went along when I got here. I found things that worked for me. I, for many years, had kind of a traditional style where I got up and talked and showed PowerPoints. And what I've observed over the years is how teaching as a discipline has grown in importance on this campus. And I now feel like a dinosaur. I have younger colleagues, particularly our instructors, who are just unbelievably good teachers. They study it like a science. So I've now become the student again, and I go to them for advice, try to learn how to do different things besides just get up and talk in front of PowerPoints. And it's actually made it fun again and much more interesting. And my teaching reviews have started to increase. So my student reviews. So it's all good all the way around. So yeah, I've really witnessed a sea change in how teaching is treated as a discipline on this campus.
Yeah, there's still just so much more to learn, right? And it's good to be a student again. That's fun. How has your science, how has the lab and your experiments and your lab technicians changed during COVID? Like, what is the lab environment like right now?
Yeah, that was tough. We were going, full bore, and then last March we just closed the door and walked away. And I had this great team of undergrads, their names were on that second to last slide, who were still coming in and feeding the snails and cleaning their tanks, albeit, not overlapping and wearing masks back when the university was shut down. And we have slowly ramped up since the university's opened a little bit, but it's still hard because Stephanie and I, we're still working largely from home. We've got kids in school on Zoom. It's actually been a real challenge. We're slowly starting to work back in. Balout's been able to go in. He also has kids at home. We just hired a new faculty RA, Vanessa Smilansky, who has just showed up like just a couple of weeks ago, and she's now getting in the lab full time. So things are really starting to pick up. So it's actually much more fun in there. It was pretty bleak for a while. I'd come in like once every other week and it'd be all dark and there'd be nobody there. So yeah, I'm really looking forward to things changing.
Yeah, I think we all are. I think I've got one, I think perhaps our last question, maybe not, but Charles Criscione is here on the call, and he is asking, what do you think the next major tech advance is needed to advance our understanding of parasite evolution itself?
Wow, it's so hard to predict. I mean, these next-gen sequencing technologies are just mind-blowing. I mean, now all these long-read technologies, there's like this little DNA sequencer on a chip that you plug into a laptop, and it'll do tens of thousands of base pairs in one read. I mean, it's just mind-blowing. Pretty much, I think we're going to soon be doing population genetics using whole genome sequences on hundreds of individuals. I mean, right now it's almost laughable. Stephanie, now we have these different individual snails that are interesting. I'm like, yeah, go ahead, sequence its genome. We have this PacBio system that we'll get like these super nice whole genome assemblies for like $2,000. It's crazy. So it's really hard to predict. Really, what the limiting factor is math. It's biocomputing and our ability to handle big data. And I can now only do this stuff with the help of people who know how to do that. And really, I think that's where all the action is in moving forward with these techniques. And 10 years from now, when I'm looking back, who knows what'll be possible. So
well, I just want to thank you again. If we were all in person, you'd be hearing another round of applause, but you'll have to take my happy smiley face as a big thank you for your time and all that you've shared. I also want to thank the Gilfillan family for making this possible in the College of Science. And thank you all so much. Have a great evening.
Thank you.
Thank you.